Hello, everyone. Good afternoon. Welcome back. Thank you for joining us. We are glad that you're here. I'm Steve.
And I'm Scott.
And together we're Backyard Musings, the Science and Technology channel.
Yeah, and again, we're talking this afternoon about another health issue, science-related health issue. So there we go.
I got to get my notes up here. Sorry, I should have that. A needle-thin, flexible brain implant is giving researchers a new way to interact with multiple brain regions simultaneously. A new type of brain implant is offering researchers a different way to interact with neural circuits across multiple depths of the brain. The device is a microfluidic Axialtrode, and they're calling it short mAxialtrode, a long and extremely thin electrode designed to place functional contact points along its length rather than concentrating everything at a single tip. So I wonder how they get past the skull if this is a long thing. How do they get through? Do they have to drill and then set it in? This approach allows scientists to record neural signals while also delivering medication with high spatial precision to different brain regions. Would you like to know more?
The work was carried out by researchers from DTU, the University of Copenhagen, the University College London, and other institutions, and has been published in the journal Advanced Science on January 21st of this year. At present, the mAxialtrode is intended mainly for basic neuroscience research. Many brain processes depend on activity that spreads across layers of tissue, making it difficult to study them using tools that interact with only one location at a time. By accessing multiple depths simultaneously, the new implant can help researchers investigate how signals propagate in conditions such as epilepsy and in functions related to memory and decision making. So maybe Alzheimer's, dementia?
Maybe.
Yeah.
The research team also points to longer-term possibilities in medicine. One potential direction involves combining targeted drug delivery with electrical or light-based stimulation in specific brain regions. Using a single implant for both functions could make it easier to study how chemical and physical interventions influence the same neural circuits.
Postdoctoral researcher Kyung-Yang Su, I hope I'm pronouncing that right, probably not, who developed the mAxialtrode concept together with associate professors Christos Markos, explains that the goal was to integrate capabilities that are usually spread across multiple devices. Bringing these functions into one implant can reduce the amount of material placed in the brain while improving experimental control and precision.
Quote, most current brain implants are based on hard materials such as silicon, which can irritate the brain and trigger inflammatory reactions in the tissue. The new implant differs in that it is made of soft, plastic-like optical fibers and has a specially angled tip that makes it smaller, and reduces the damage caused when it is placed in the brain, says Kunyang Sui.
He adds that the technology is still at an early stage with extensive testing. Further development and regulatory approvals required before any clinical use would be possible.
Okay. Many neuroscience experiments today rely on conventional flat-end optical fibers. These thin glass or plastic fibers are commonly used to deliver light deep into the brain, including in optogenetics, where light is used to activate nerve cells. Their main drawback is that their influence is limited to a single point.
The outermost end of the fiber is known as the distal tip, or the nose of the fiber. Because all light emission and contact with brain tissue occur at this location, researchers can usually stimulate or record activity in only one brain layer at a time, even though many important brain functions depend on interactions across multiple layers and deeper regions.
The needle-thin mAxialtrode is manufactured using a process in which a large polymer rod is heated and drawn out into a very thin fiber. The process can be compared to making sugar thread, only much more precisely. In the middle runs a core that conducts light. Around it are 8 microscopic channels that can carry fluid and also accommodate very thin metal wires for electrical measurements.
The fiber is less than half a millimeter thick and is so flexible that it moves with the brain instead of cutting through the tissue. The difference in stiffness is important because hard implants often trigger inflammatory reactions in the brain over time.
This is pretty cool. So they're not penetrating brain matter; it just goes with the flow.
Yeah.
The researchers have not only tested the technology in the lab, but also in vivo, that is, in mice. Here, the brain electrode was implanted in the brain and connected to light sources, measuring equipment, and small pumps for fluid supply.
The experiment showed that the researchers could stimulate nerve cells with blue and red light, measure electrical activity simultaneously from both superficial and deeper brain layers, such as the cerebral cortex and hippocampus. I guess I'm pronouncing that right. And inject different substances at different depths up to almost 3 millimeters apart. All examinations and simulations could be performed with a single lightweight fiber and that the animals could carry without any obvious signs of discomfort. That's interesting.
Yeah, so this kind of, while we're reading this paragraph here, this kind of reminded me of the novel Frankenstein, how the guy was trying to build from dead body parts, reanimate flesh. And so this kind of reminded me of it. The in vivo experiments in neurophysiological validation were carried out in close collaboration with Associate Professor Rune W. Berg from the University of Copenhagen and Associate Professor Rob C. Wykes from University College London, who contributed expertise in neural circuit analysis and epilepsy-relevant models.
The researchers behind the brain electrode are in the process of patenting the underlying technology and clarifying the possibilities for testing the electrode on patients in the clinical department. So, sounds promising.
Well, something different.
Yeah, because I mean, that's a tough area to be operating on, right? It's just it's not a routine thing when you start getting into the brain.
Yeah.
Very interesting. So, okay.
Well, we hope that you do something nice for your sweetheart today on Valentine's Day, and we hope you enjoy the Sabbath tomorrow.
Yep. Enjoy your Valentine's Day. Don't forget your significant other, and enjoy your Sabbath. Take care. We'll see you on Monday.
See ya.